Good afternoon, friend. Today I have decided to discuss a very unique topic of from principle of food science and nutrition. Under this topic, I have decided to discuss the very important nutrient because till now we have completed the discussion on carbohydrate and lipids. Now today I have decided to discuss on the protein. And protein, as you know, that it's as a very vital and very important in nutrient in the nutrition, and which is the very essential thing. And the protein is not one of the most important thing about protein is that protein is not stored anywhere else in the body of the human being. So anyway, now I am sharing the clips with you, please, and then we'll have a discussion simultaneously. So protein, in this topic, we'll discuss the importance of protein and classification. Myself, Dr. J.K. Sharma, I am from School of Agriculture, and I had this vital topic I have decided to discuss with you. And the objectives of this topic is that what we'll learn finally when we'll have a discussion on the protein, the concept and the importance of protein in human life and in the life of any living organism, then the type and the characteristics of and the specialties of protein that these are the things those we'll learn, and finally when we'll have a discussion we'll complete our discussion. So looking to these both of these things, I have decided to discuss this topic which is of very much important, and so I need your 100% attention in during the discussion. I'll repeat time and again that the same thing which I have discussed once. So proteins are nothing. They are the combination of the amino acids, and they are the basic unit of the. It means the chain of amino acids being called as a protein, and the amino acids because it contain one amino group and another carboxyl group. Both groups they are there. So that's why in general the formula of amino acids NH2 CHR COH. It means here the both the ammonical group and the carboxyl carboxyl group is also there. So both of these atoms they are attached with the in the amino acids, and they are attached with the CONH bond, and that is why the ring is like that, and that is why it's a chain of amino acids. So carbon atom at which both functional group are bonded is known as the alpha carbon atom, and such amino acids are known as the alpha amino acids. So the carbon atom at which both of the functional groups are bonded is known as a alpha carbon atom, and such amino acids are known as the alpha amino acids. So once again I repeat that the proteins are nothing, they are the amino acids basically, and they are the units of the amino acids. It means in a chain if we have amino acids, that if one amino acid added, one amino acid reduces, on the basis of that we have given the name of these amino acids. And then because of contain one amino acid NH2 group and another carboxylic group COH group, the general formula of amino acid is H2 and CHR COH. So here the carbon those when the carbon atom at which the both of the functional group are bonded is known as the alpha carbon atom, and such amino acids are known as the alpha amino acids. Here we'll see that the abbreviation of amino acids, that the name of the amino acid, that alanine, and that the one letter abbreviation A, arginine R, aspargine ASN, aspartic acid D, cysteine C, glutamine GLN Q, one letter abbreviations. Through this one letter we are also giving the name of the name was hidden through the abbreviation. Sometimes we are addressing the carbohydrates, so we are addressing the amino acids. Then we have glycine GLY, histidine HIS H, and there's isoleucine IS ILE. Then I is there, then the leucine L only is there, then lysine LYS, and K is the one letter to we are abbreviation with in the abbreviation methionine, MET and M, phenylamine and PHE and F, proline P and Prioro, serine SER and S, threonine that THR and T, tryptophan that TRPW, triosine TRY and TYR and Y, valine and asparagine and aspartic acid that B, and the glutamine and glutamic acid CHAD. So these are the abbreviations. Those we have to understood that what is the meaning. That the instead of writing a full name, sometimes we write in the form of the abbreviation in the one letter. That the we have understood that these are the amino acids. Those they are having the name. Only the name we will write, then with O. Then the classification of amino acids. Amino acids are classified into different ways based on the polarity and structure. Polarity means what kind of that the arrangement is there in the chemical structure. On the polar, that the bonding, what kind of bonding is there. On the basis of that the classification, then the structural classification, then the nutritional requirement. On the basis of that the amino acids, they are classified among the different categories. So that to be discussed over here in detail. That to be discussed over here in a detail, and we have to understood this thing, that how we are discussing the how we are classifying the amino acids. So amino acids, they are classified on the basis of the polarity, that what kind of that polar bonding is there, then the structural, that chemical structure and nutritional requirement. These are the basis of the classification of the amino acids. Now, based on the polarity, amino acids are classified into four groups: non-polar amino acids, polar amino acids with no charge, polar amino acids with positive charge, polar amino acids with negative charge. So here the four type of that classification is there based on the polarity. It means non-polar. It means there is nothing like that on the polar. That's the and then the polar amino acid with no charge, then the positive charge, then the negative charge. And the fifth one is amino acids are classified based on the those non-polar R group, unchained polar R group. This is also one of the classification. So charged polar R group, the polar side chain amino acid B are called hydrophobic, and amino acid with uncharged being called as a hydrophilic. So if charge is there, then the hydrophobic, and if charge is not there, it means the hydrophilic. So that kind of that the fifth class, five type of classification is there, and I'll repeat time and again that the what kind of that classification is there. So this is very important that So based on the polarity amino acids, they are classified into the four groups, and the fifth one is also that the amino acids are classified based on those with non-polar R group and uncharged polar R group, and then the charged polar R group. The amino acids are called hydrophobic, and the amino acids with uncharged polar bonds are the hydrophilic. So that these are very important things that we have to understood this thing, and we have to take care of these things with us. So these all are very important things that why we have that due to the interruption. Sorry for the interruption because it is was activity lost the connection. Now when I am sharing this with you, and I hope that now it is quite visible for you, and you will have this kind of thing in front of you. We have that non-polar amino acids. They are there, and we'll have a discussion on that. That these are the non-polar that acids. They are there, and that non-polar amino acids hydrophobic aliphatic beta. So non-polar acids have equal amino acids and carboxyl group are equal neutral. These amino acids and have no charge on the group. The hydrophobic, it means there is no charge on these groups. That is also very important when we are discussing them. The amino acids in this group are alanine, valine, leucine, isoleucine, theanine, lysine, tryptophan, methionine, and proline. These are the amino acids, so they are having the charge on their R group. That's why we are calling the hydrophobic amino acids, and they are also being called as a hydrophobic amino acids aliphatic R group. Non-polar have equal number of amino and carboxyl means both are having the same equal number. That's why they are neutral, and because of that neutral, there is no charge on them on our group, and that's why they are being called as a hydrophobic. Hydrophobic, it means there is no charge. And these under this group we have a few valine, leucine, isoleucine, valine, glycine, tryptophan, methionine, and proline. These are the important amino acids of this group. Then valine, leucine, isoleucine, and are important in promoting hydrophobic intrinsic protein structure. So that we are also observing over here that this is the equation that is very important from that point of view. These few things you can see easily that is the both numbers equal. That's why there is no charge on then that's why they are calling that the side chain of are important in promoting hydrophobic interaction with protein structure. So then these are the structural presentation of the all the important aniline The abbreviation also G we have written over here, alanine A and the valine V, and then leucine L, and then isoleucine I. So these are the abbreviation, and these are the chemical structure of the amino acids of the hydrophobic nature. It means there is no charge. Then here we'll have a amino acids, those who are having the, those who are having no charge, they are having no charge, they are having no charge. They are having no charge. This is also very important thing, that what kind of situation is there. So we have to look after these things, and because of the poor net connectivity, I am not able to share the screen with you. I am trying that. I'll do it very early, and I am sure that very soon I'll do it. Net connectivity is not there. I'm just waiting that. I think that there is a poor net connectivity. That's why Here is I'm able to. I think because of the poor net connectivity, I am not able to share the screen right now. I'm just waiting within 2 minutes. If it will correct, then we'll have a discussion on that. So I'm just trying to share the screen with you. I'm just trying to. I'm trying to share this, but I'm share this thing with you. So once again, bear with me. I'm just trying to this thing time and again, but unfortunately, I think that there is still with technical. That's why I am not able to share the thing. So you just wait. I am just trying to. I'm just once again trying to correct the things. So here, once again, the connection loss is there. So anyway, I am trying to connect once again. You please wait and bear with me. I hope that very soon it will be started. Recording will be started. So we have a discussion on that entire routine we'll discuss, and we'll have a discussion on that we will know that what kind of that situation is there. So I'm just once again trying to. I'm trying to share the suite, but I think it was not allowed by. Now it is fine. I'll share with you. So now it is visible, and now I am sharing this thing with you. That based on polarity, amino acids are classified in four groups as follows: non-polar amino acid, polar amino acids with no charge, polar amino acids with positive charge, polar amino acids with negative charge. Amino acids are classified those based on the based on the those non-polar R group uncharged or R group charged polar group. The non-polar side chain amino acids are called hydrophobic, and the amino acid with uncharged polar side chain is called hydrophilic. So charged one is called hydrophobic, and uncharged will called as a hydrophobic. So that we have discussed, and now we will have a non-polar amino acids and hydrophobic aliphatic R group. Non-polar amino acids have equal number of amino and carboxyl group and are neutral. These amino acids are hydrophilic phobic and have no R group. The amino acids in this group are alanine, valine, leucine, isoleucine, phenylalanine, glycine, tryptophan, methionine, and proline. These are very important. And here we have the structure of these amino acids. Now you'll see that the screen will change very soon at your end. So these are very important, that the few these are the few important that amino acids, those who are very important, and they are sometimes calling this thing, and we are sharing this thing over here. So now you'll see that the chemical structure that the both are having amino group and the carboxyl group, and they are in a equal amount. The side chain of alanine, valine, leucine, isoleucine are important in promoting hydrophobic. Hydrophobic means uncharged within protein structure. Protein structure. Now the polar amino acids charge. These amino acids, they are very important. They are also very important that they do not have any charge on R group. These amino acids, they are not having any charge on amino group, and they are also very important. They are also very important that they are also very important that these are the amino acids do not have any charge on R group. These amino acids participate in hydrogen bonding of protein structure. The amino acids in this group are serine, threonine, tyrosine, cysteine, glutamine, and asparagine. These are the few important amino acids of this group. Amino acids of this group, they are very important. They are very important. So that's why we are discussing over here to them. Now the amino acids, these amino acids do not have any charge on the R group. These amino acids participate in hydrogen bonding of protein structure. The amino acids in this group are serine, tyrosine, cysteine, glutamine, and asparagine acid. So these are a few important. Those polar amino acids with no charge. With no charge, it means here there is no charge. The structure will see also. the structure will see also over here. That is also very important. That structure will see that this is a structure which is very important. That to be discussed over here. That what kind of that structure which we have. The polar uncharged group, that the serine first one, then the asparagine, then the glutamine. It means there is no charge. Everything is balanced and there is no charge. That's why the group is very different and distinguished. Then aromatic R group, phenylamine, tryptophan, these are also very important. They are also not having any charge. Polar amino acid with positive charge. Now we'll see that the polar amino acid, those who are having the charge of positive. Polar amino acid with positive charge have more amino acid as compared to carboxyl group, making it the amino acid with the have positive charge on R group are placed in this category, and they lysine, arginine, histidine. They are like lysine, arginine, and histidine. See that here the basic this group include lysine, arginine, and histidine. As is then the lysine, Ly, and then these are few important that thing, the chemical structure which is in front of us, and which is also very important that we have to understood this thing, and that is the chemical structure of these no charge. Then the polar amino acid with negative charge. Here the first one we have discussed, those who are having positive charge. Now we have the amino acid those who are having negative charge. Negative charge. It means the polar amino acid with negative charge have more carboxyl group than amino acid group, making them acidic. The amino acid which have negative charge on R group are placed in the category. They are dicarbo amino acid because they are having two carboxyl group with them. They are aspartic acid and the glutamic acid. They are the aspartic acid and the glutamic acid. Negative that R group chemical structure aspartate and glutamate. The two amino acids with acidic R group are aspartic and the glutamic acid. These are very important that we have to understood that what is the charge, what is no charge, what is positive charge, what is negative charge. That on the basis of that we have a description of the amino acids. Classification is the availability of the amino acids. Classification is based upon the availability of the amino acids. So this classification is like that. There are two types of amino acids: essential amino acids and the non-essential amino acids. So essential amino acids are that cannot be synthesized by the body. Example: leucine, lysine, lino. These are important. That non-essential acids are that can be synthesized by the body, and there are 11 non-essential amino acids. Example: alanine or like that. So we have to understood that on the basis of the need. If they are essential because they are not being produced inside the body of the human being, that's the human being or animals, those they have to produce, they have to take in the diet. And the non-essential, those who are produced by the body, so there is no need. For example, the alanine, the first one is the leucine. Then here we have essential. The first one, the histidine, isoleucine, leucine, lysine, methionine, phenylamine, and then the threonine, tryptophan, and valine. These are the essential amino acids. Those who are not being produced inside the body of the human being. So it was necessary that they have to incorporate in the field. Then the second side we have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. These are the amino acids. Those who are also important, but they are non-essential because body itself produces these amino acids inside the body. So they are non-essential amino acids. So we have to understood this thing that whether the is essential or non-essential, that we have to think very closely. Now the deuterium, it's a very important thing that when the amino acid is dissolved in water, it exists the inner salt carrying both positive and negative charges. This occurs of this association or carboxyl group to release hydrogen ion, which passes from carboxyl group to amino group. The acids possessing both positive and negative charges are called deuterium. The amino acids, those both positive and negative charges with them. It means the neutral that amino acids, those who are having both positive and negative charges, they are being called as a deuterium. Since it is having both the charges, positive and negative, the net charge of deuterium is always zero because both are having the both charges they are having with them. So that's why the generally the zero deuterium charge is net charge of deuterium is zero. Then the amphoteric nature of amino acids. Amino acids are amphoteric compounds as they contain both acidic and basic groups. In acid amino acids carry positive charges. In alkaline amino acids, they carry negative charges. The point at which the deuterium has no net charge is called isoelectric bond. It means the quantum point where the point at which the deuterium has no net charge. It means generally the deuterium we know that the both negative charge and if the total is zero, that is being called as a deuterium. At the point which the deuterium has no net charge is called as a isoelectric point. It means that the zero is charge that to be called as a isoelectric point. Now the properties of the amino acids. This is also very important that we can have the property of the amino acids are white crystalline, that physical property, the white crystalline substances. Most of them are soluble in water and insoluble in non-polar solvent. It means amino acids just like they are not like the fatty acids. They are solved in the water. They are not insoluble in non-polar organic solvents like ether, that the chloroform, benzene, like that. And the aliphatic and aromatic amino acid part, particularly those having several carbon atom, are limited solubility in water, but readily soluble in. But those who are having the particularly those who are having the several carbon atom have limited solubility in water, but readily soluble in solar organic solvent. They have high melting point, varying to 300 degrees centigrade or even more sometimes. So amino acids they are having they melt at a very high temperature, that's more than 200 degrees in general, and even more than that 300 degree also. They are tasteless, sweet or bitter. Sometimes they are taste of these amino acids, but sometimes they are tasty, they are sweet and they bitter sometimes. So these are the physical property of the amino acids. So we have discussed in detail that what are the amino acids, how we can have the idea about the amino acids, and how we can discuss amino acids in detail. So we have that now. We once again I will try to discuss the deuterium. When an amino acid dissolved in water, that they are having both the type of that we know that negative. So the amino acids possessing both positive and negative charge are called deuterium. And the isoelectric point is also very important. That that is also very important. That at point where the deuterium has no net charge is called as a isoelectric point. Means when the deuterium there is no charge, that point is called as a isoelectric point. So we have discussed a lot of things here, and I hope that through this discussion, we'll try to get the right message about the protein that we have discussed. It's the chain of the amino acid that we have discussed, and we have finally we have that the properties of amino acids. Acids are white crystalline substance. It means they looks like white crystalline also, and substances. So they are like that, and most of them they are soluble in water. They are insoluble in the non-solvents like ether and chloroform and benzene, and they are but they are few. Those who are having the several carbon atom have limited solubility in water. They are readily available and soluble in the non-polar solvent like that ether and benzene. So this is very important about the amino acid that we have discussed over here. They have high melting point also, that more than 200 degrees centigrade. Generally they melted at more than 200 and even more than 300, 400 degrees centigrade. Sometimes some amino acid they melted over there, and they are tasteless. Some few of them they are tasteless. Some they have the sweet taste. Sometimes they have fewer of them. They have the bitter taste. So taste wise also we can have that idea that which amino acid is there. And finally we will conclude that the amino acid, the protein is nothing. It is just the chain of the amino acid that we have discussed over here. So in this topic, I have tried to elaborate the protein as a unique nutrient which is integral part of the genetic bond. You know that RNA, DNA, they are of protein actually. So that's why it's very important that the protein supplementation in the body is very much required. And one of the most important quality of protein is that they are not being synthesized, and they are not being stored in the body. So every time whenever required, they are to be with the chain, because the chain of the amino acids that is available in the body, and they are as per the need of the body. They are to be converted into the desired protein structure. So that is, and the few of them we have discussed, that those who are non-essential, it means they are being produced inside the body of the human being. So they are not that much of importance. But few they those who are very important, because they are not being produced. So for all of them, you see that the essential histidine, isoleucine, leucine, lysine, methionine, phenylalanine, and then the threonine and the tryptophan and valine. They are the essential amino acids. It means you have to incorporate both of these amino acids into the feed or into the food which we are consuming. Then there are alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, cysteine, proline, serine, threonine. These are not that much of importance. These are not that much of importance. So we can avoid it. Sometimes if it is possible, we'll avoid them and we'll not incorporate them into our regular diet. So these all about the protein that we have discussed over here. And now I'm just going to all the objectives that we have concept, we'll discuss, and we have discussed the concept of protein, and we have also classified the protein on various basis, on various parameters, and then we have also the characteristics we have also up to a certain extent. So I think that we have completed this topic, and anyway, if you have any query, you please ask. If you don't have any query, then it's okay. Otherwise, if you have query, you please send into the chat box. So thank you very much for joining with me, and have a nice day.